Symposium about Affymetrix, so I leave the platform and leave it to the representative of Affymetrix, I believe.
Hi there, so my name is Fiona Togneri and I'm a clinical scientist from the West Midlands region in the United Kingdom. I'm here today to talk about our experience of using Affymetrix platforms. So just first of all, a little bit about who we are. So the West Midlands Regional Genetics Laboratory is a fully comprehensive genetics service. We provide regional genetic diagnostics, and we also have quite an interest in translational research. We run approximately 55,000 samples per annum across a variety of different reasons for referral. We have approximately 150 laboratory staff and also clinical genetics, so consultants and counsellors that have patient-facing roles. We also have a strong research programme, and we in fact have shared facilities and also staff with the University of Birmingham, which is situated on the same site as our diagnostic laboratory. So we are based right here at the centre of the United Kingdom, and we cover a population of approximately 5.6 million people. It's quite a diverse population with several different ethnicities represented, and this can make for some interesting and challenging genetics for us. So within our microarray technical hub, we run a variety of different microarray assays, each of which sort of feed into the different germline and cancer programs within our laboratory and the 4 different separate patient pathways, which I'll discuss individually as I go through this presentation. So in order to identify genomic imbalances in these patients, one of the arrays that we use is the Cytoscan HD array. So it's a DNA copy number array that has both polymorphic and non-polymorphic oligonucleotides on it. We also run the AML profiler array, which is an RNA expression array which allows for the stratification of patients with acute myeloid leukemia. In addition, one of the platforms that we're quite excited about just now that we've recently started using is the Affymetrix OncoScan array. So this again is a DNA array that allows for multiplexing, so identification of somatic mutations and also copy number imbalances, and I'll discuss this in more detail as I go through. And we also run other platforms by different manufacturers, but I'm going to concentrate on talking about the Affymetrix ones today. And in particular, I'm going to concentrate on talking about the Cytoscan platforms and also the OncoScan. So the Cytoscan platform provides a really useful platform for investigation of patients in different patient pathways, and we've actually utilised a Cytoscan array in all 4 different patient pathways within our site. So we've used this platform using DNA extracted from a variety of different sample types, and we've really found that we get consistently good results. So developmental disorder genomics is the main field of genetics where copy number arrays are currently utilised and are known to have very clear clinical utility. As I'm sure most people in this audience will know, able to identify copy number imbalances in up to 20% of individuals with normal cytogenetic findings. So the Cytoscan array, we feel, provides a very useful platform that adds more to our diagnoses than we're able to get from other platforms that we've utilised in the past. And I'm just going to try and illustrate this now with a couple of patient examples. So patient 1 is a 7-year-old girl who was referred to us with developmental delay. She had agenesis of the corpus callosum, microcephaly, optic nerve hyperplasia and short stature. We ran this patient on our ISCA 60K oligonucleotide array. So this is an Agilent-based platform. And we were able to identify that this patient had a deletion somewhere between 24 kb and 146 kb in size. So this is a massive difference. Over 100 kb difference between the maximum and the minimum size of the deletion that may be present in this patient. And the reason for this is due to the very gene-centric positioning of the oligonucleotide probe. So you can see, do I have a pointer here? So you can see there's a huge gap here between the minimum deleted point and the maximum deleted point. So what size is this patient's deletion? We then ran this patient on a Cytoscan 750K array, and actually we were able to identify that the deletion is 26 kb in this patient. And this was sized identically in replicate, so we ran the array twice and twice got exactly the same results. The deletion includes exons 2 to 9 of the PDHA1 gene, which encodes a subunit of the pyruvate dehydrogenase complex. Patients with haploinsufficiency of this gene have a variable clinical presentation, and this is due to residual enzyme activity, especially in females, due to different levels of exon activation. We have now shown that this patient has reduced PDH activity in fibroblast studies, and this work was undertaken at the University of Oxford in the United Kingdom. Patient 2 is a 1-year-old girl. She had a 46XX karyotype by cytogenetics. She had microcephaly, microphthalmia, poor vision, a squint, and truncal hypotonia. Microarray analysis in this patient was able to identify that she had a mosaic duplication on the long arm of chromosome 1. So this trace here shows you the non-polymorphic oligonucleotides, and you can see these are slightly shifted up. But where this becomes really apparent is within the SNPs and on your smooth signal here, which takes the information from both polymorphic and non-polymorphic oligos and is able to show that in fact this is copy number of 2.3, so it's not duplication in all cells, not copy number 3, it's somewhere between the two. This duplication is actually 25 megabases in size, so absolutely massive, something that should clearly be cytogenetically visible. However, this was not identified by cytogenetics, and even once we knew which aberration was present, we knew which bands were involved, and we went back and looked at the cytogenetics, we weren't able to find this in our synchronized T cultures. And this is likely simply due to the fact of cultural selection and variable cell populations that are present within a blood sample, and this is something that's well documented in the literature. So when we requested a repeat sample from this patient, we undertook fish and uncultured preparations, and indeed we found this duplication present in approximately 46% of interphase cells. And this really shows the benefit of SNPs there, so the SNPs help you identify low-level mosaicism at a more detailed level and help you to assess exactly what percentage of mosaicism you're talking about. Patient 3 was prepared with an osteogenesis imperfecta-like phenotype. However, it was an extremely severe phenotype with very pronounced clinical features. This patient was from one of our consanguineous communities, and you can see the karyotype here. The purple lines on the karyotype here indicate regions where the patient's genome is homozygous. In fact, she had approximately 8.3% autosomal homozygosity, and this is consistent with cultural consanguinity and likely parental first cousins. So, of course, with such levels of homozygosity, there's risk of recessive allele mutation expression, and in fact, this patient had LOH for much of chromosome 7, and this involved one of the two main osteogenesis imperfecta genes called 1A2, and in fact, this patient was homozygous for a mutation in this gene, which is normally associated with an autosomal dominant condition. However, this patient had a severe phenotype due to rare autosomal recessive severe osteogenesis imperfecta. And again, this is the benefit of the SNPs present on the platform that allows you to identify these regions of homozygosity that would not be able to be identified if you were looking at a simply copy number only array. And to show you the other side of this, so as you can see, this patient's genome in comparison to the patient we showed you earlier on does not have much homozygosity, but they do have homozygosity for all of chromosome 15. And this was a 3-month-old child with neonatal hypotonia. And this is in fact maternal UPD for all of chromosome 15, diagnostic of course of Prader-Willi syndrome. And again, it's just the benefits of the SNPs on the platform. So yes, there's SNPs on the array, but lots of arrays out there have SNPs on them these days. So what else did the Affymetrix platform give you? Well, something that we find particularly useful is the fact that it is a whole genome array and not as gene-centric as some of the other ones on the market. So the ISCA targeted microarray platform is currently employed by many genetic diagnostic labs, certainly in the UK and across a lot of Europe at present. However, the ISCA gene list is now accepted as being less than ideal and actually really quite outdated. The International Collaboration for Clinical Genomics, formerly known as ISCA, have recently undertaken an evaluation of all of the genes present on their platform themselves. So there are 382 genes that are specifically targeted on the ISCA 180K array design. And ISCA have shown themselves that 46% of these genes only have sufficient evidence for haploinsufficiency, which is generally what you're looking at on a copy number array. And actually 34% of the genes have absolutely no evidence for haploinsufficiency at all. In addition to this, of course, there's going to be many non-ISCA genes, which are now known to be extremely important, but the limited of these genes on your platform is extremely limited. And this is something that's being addressed currently by ISCA themselves as part of the ISCA dosage sensitive map project. And this is a rapidly advancing field, however, and continual updates are likely to be required. So it's okay to update this now, but you need to continually update, and therefore you need to be willing to continually release different microarray platforms. And is it not better, therefore, simply to target all of the genes in the genome that are known protein coding genes, and then alter your targeted analysis continually based on current literature? And this is something that's possible with the cytoscan array. So I just want to illustrate this further point by discussing the 5q14.3 microdeletion syndrome. So this microdeletion syndrome was first described in 2009. The MEF2C gene is the main candidate gene within this microdeletion syndrome. Patients with haploinsufficiency of this region have a severe Rett-like phenotype. They have severe learning difficulties, absent speech, limited walking, severe hypotonia, seizures, and a similar facial gestalt to each other. And this is 4 of our patients that we've identified so far with microdeletions in this region. And all of these patients were identified at a time when we were using this Agilent ISCA-based array. 2 out of 4 of these patients, however, have just 3 oligos deleted within their region, and 3 oligos is actually the minimum number of oligos required in order to call an imbalance in this array, and that leads to anxiety then, because have we missed smaller intergenic exonic deletions in patients with this syndrome? So MEF2C is not an ISCA gene. There's between 3 and 10 oligos covering this gene in each of the different ISCA designs. So this, just to illustrate it, is what these 4 patients look like when they're run on a cytoscan HD array, and you can see the vast number of oligos that are included in each of the patient's deletions. And if you look at the first patient at the top here, you can see that actually this shows that she's 2 separate deletions, something again that just wasn't identifiable with the other platform. And this just shows the vast difference here. So you can see even in the patient with the very smallest deletion, there's 168 probes now covering that deletion as opposed to the 6 that we had previously. Now MEF2C has now been recommended for ISCA targeting, but it's still not targeted on any of the ISCA platforms that are currently available on the market. So moving on from developmental disorders then towards reproductive medicine. So microarrays are being utilised more and more in this setting for prenatal diagnosis and also fetal pathology. And we've used a cytoscan array using DNA extracted from amniotic fluid, from fetal blood, from CVS and from fixed cells, and in all instances gained really informative results. And just to show you one example of that was a prenatal diagnosis for a fetus which was at 28.5 weeks gestation. So this is really quite late gestation, and the later on in gestation you go, the poorer the quality of the DNA that you're able to extract from the amniotic fluid due to the other contaminants that are present. It was 46XY by cytogenetics, and QF-PCR was normal. Due to these other contaminants and things present, we were only able to obtain 32 nanograms of DNA. Now that's just 12.8% of the amount of DNA that Affymetrix recommends that you apply to this array, but it's all we had in its prenatal diagnosis, so we gave it a go. So this shows the results from that. So running the cytoscan 750K array, analysed using the default high-resolution setting on the platform. And we identified 5 calls. 4 of these could be easily, very quickly disregarded as being either common microarray artefacts or regions of benign CMV. But we did have 1 pathogenic CMV, and that's this deletion here. So it's 112 kb in size, and it's a microdeletion in the Rubinstein-Tabby syndrome region. At 16p13.1, including the CREB-BP gene. And this is consistent with the reason for referral in this patient, as VSD is a recurrent finding in these patients. And this just shows that small deletion there, as you can see, slightly disrupting CREB-BP here. And again, this is the benefit of using an array platform where you're able to utilise suboptimal quality DNA as you really can with the cytoscan arrays. Hematological malignancies is another huge growth area for microarrays at present, and we've used this DNA for different hematological malignancies. We use the 750K standardly for patients who we're diagnosing with MDS and where their clinician wishes us to obtain more genomic information. To give an example of one of the patients in where microarrays has been useful so far, I'd like to discuss one of our patients who presented with acute promyelocytic leukemia. So this patient had APML and they unfortunately had extramedullary relapse in the skin, something which is unusual. So presentation, this patient had the standard PML-RARA 1517 rearrangement that's present in these APML patients. They also had additional chromosome 21 and all abnormal cells present, and there was an inversion of chromosome 11 and also a different subclonal population. At relapse, a paraffin embedded tissue section was sent to us. We were able to do interphase FISH in this and show that the RARA gene rearrangement was present in 30 out of 155 cells, but we couldn't do cytogenetics, so we didn't know which cell line had relapsed. So we were able to get 217 nanograms of paraffin-embedded tissue DNA and applied this to a cytoscan, I believe, HD array. So you can see we have this gain of chromosome 21 that was present in all of the cells. It's been called here on the Affymetrix software as many small duplications, and that's simply because it's a mosaic that's only present in a subpopulation of cells, and actually since this case has been run, there is an improvement to the software which actually calls these mosaic deletions nicer than that. And there's also a gain of the terminal region of chromosome 13, and there are two tiny mic deletions present exactly at the inversion breakpoints that was identified at cytogenetics. So the gain in chromosome 13q is really interesting. Sorry, it was the 750K array, that corrects me there. The duplication, you can see, is of this region here, and actually it's copy number 4 as opposed to copy number 3, so indicating perhaps a triplication. But if you look at the SNPs at the bottom here, you can see that it's entirely homozygous, so these, your AB heterozygous SNPs are present along the centre call here, and you can see that this region is entirely homozygous, so what this tells us is that in fact this is 4 copies of the same genomic region with loss of the other allele. So this tells us that it was our most developed cell line that was present in our extramedullary relapse, that this cell line has further developed as there was addition of this 13q imbalance, and we have since run the presentation DNA, and this was not present at presentation in this patient in any of the cell lines. And again, this is just a benefit of an assay that allows utilization of suboptimal quality DNA, but also the inclusion of the SNPs here is really important to be able to tell us exactly what was going on at 13q. So FFPE, as I'm sure many of you know, can be extremely suboptimal, and for this reason, Affymetrix have actually recently released an alternative specific platform for this. Excuse me. And this is called the OncScan array, which I'll go on to talk about in just a few moments. First of all, I just wanted to sum up what I was saying about the cytoscan array. So, of added value, you have the resolution of the array, the fact that you can identify imbalances down to the exonic level, 1 kb spacing of the probes. You're able to do really good software masking, which is more flexible than most arrays. So this means that you can look at all of the genes, and okay, in, for example, a prenatal diagnostic setting, you might not want to know about all of those genes, but what you can do is tell the software about the genes that you want to see. So basically create your own targeted array, so the only regions that you know to be clinically significant are you seeing anything about. The inclusion of the SNPs is really important. I've shown it to be important both in a constitutional germline setting and also in an acquired setting for showing regions of the genome that are homozygous, whether that be UPD or regions that are identical by descent due to consanguinity, and also allow for easier detection of mosaicism. And something that we've particularly enjoyed about the use of the cytoscan array is in fact that if we have a patient failing on our adjuvant platform, we'll use that DNA and put it on a cytoscan array because we know it's going to get us a better result. So now I'm going to move on to talking about the OncScan array. So the OncScan array, as I said, has been designed by Affymetrix specifically to allow for the use of formalin-fixed paraffin-embedded tissue DNA. And we're currently running this platform within our own laboratory as part of a TSB-funded project for stratified medicine, and I'll explain what that sentence means in the next few slides. So stratified medicine is therapy targeted towards specific stratified patient populations on the basis of a predicted biomarker as a step towards personalised medicine. So this is to move us away from the idea of treating all patients with the same type of cancer, with the same type of treatment, and to move us on to treating those patients depending on which biomarkers are there, depending on which acquired genomic mutations this patient has, as opposed to just the tissue in which the cancer has occurred. So routine tumour profiling has huge potential to optimise therapeutic efficiency and minimise toxicity in cancer patients if we can specifically target what's going wrong as opposed to simply killing all of the cells with chemotherapy. So this not only has the opportunity to save money because we're treating patients with the drug that we know is most suited to that patient, but also to save patients' lives. So the most classical example of this that most of you will probably be aware of is the wonder drug imatinib utilised in CML patients. So patients with CML have this recurrent translocation between chromosomes 9 and 22 that leads to the formation of the fusion gene BCR-ABL, which is a constitutively activated tyrosine kinase. Imatinib is specifically designed as a tyrosine kinase inhibitor to specifically inhibit this fusion gene. And there are now many examples of that available and currently utilised in current practice. So leading on from this, the Cancer Research UK, a massive charity in the United Kingdom, developed their stratified medicine programme. So the idea is to develop a cost-effective strategy to analyse tumour samples for a range of relevant DNA markers that will help predict cancer treatment. So the programme is focused on 6 tumour types, the 6 most prevalent tumours, which are colon, breast, ovary, lung, prostate and melanoma, and also in a small number of genes where there's already targeted therapies available so that patients can be immediately treated on finding these mutations. There are multiple laboratory techniques currently employed in identifying the different mutations associated in these different genes, and clearly there's a need for multiplexing. So the Technology Strategy Board, who are the national innovation agency in the UK, are currently undertaking several different collaborative studies, and one of these is with Affymetrix. So this study is ongoing to develop the OncoScan assays, a robust, reliable, multiplexing clinical tool to guide tumour therapy. So we are taking part in this study together with Almac Diagnostics in Northern Ireland and also the Leeds Institute of Molecular Medicine. Phase 1 and 2 of this study are coming to completion at the end of this calendar year. We've fully developed the assay and fully validated it, and it's been run on 200 samples in triplicate across the 3 sites. Phase 3, which is the most exciting phase, will start in February next year, and this will look at 3,000 retrospective and prospective patient samples in different clinical trials. So why do we need a specific platform for FFPE? I mean, I've already shown you a result of FFPE on the cytoscan array, and sure, the results looked quite nice, but they're not as nice as you would get from blood DNA, so really something different is required. There are millions, some publications claim up to 1 billion archival formalin-fixed paraffin-embedded tissue samples stored all over the world at present, which provides an enormous and invaluable repository for information. These samples hold a wealth of data for biomarker discovery, drug development and cancer research, so it's really important that we're able to utilise them. However, the fixation process and storage of FFPE leads to nucleic acid degradation and base modification, which means that you really do get a suboptimal quality and a suboptimal quantity of DNA out of these samples, and the OncoScan has been specifically designed to overcome this. So this is what the MIP probe looks like. So the MIP, or molecular inversion probe, is what underlies the OncoScan array. So this is what it looks like, and you can see the 2 regions of genomic homology at either side of the probe have a genomic footprint of actually just 40 base pairs of DNA, and that means that DNA can be really highly degraded. As long as you have a little over 40 base pairs of DNA remaining, the OncoScan array will be able to utilise that sample. So I just want to show you quickly how this assay works, because it's really quite neat. So the MIP probe is a neo to your genomic DNA. The AT and GC DNTPs are then added in separate reactions, able to fill this single nucleotide gap in the probe. Linear DNA is then digested, so that will be your genomic DNA, as you can see being digested here, and also any uncircularized padlock probes. So it's a little bit like MLPA. It creates a circle if it finds the mutation it's looking for only, and therefore those circles are the ones that are maintained, and the rest is degraded. The probes are then cleaved at this site here, and then PCR is undertaken of all of those probes that were able to circularize in the previous stage. The probes are then cleaved at site 1 to remove this genomic homology region, and in fact it's this green tag which started off on the probe that's then hybridized to the array. So the OncScan assay allows us to utilise highly degraded, low concentration DNA. They ask for 80 nanograms of DNA to be put into the reaction, which is an extremely small amount of DNA, but actually many publications in the literature have gone down as low as 4 nanograms and still been able to identify at least copy number information of really high quality. And as I said, there's just a 40 base pair genomic footprint required, meaning that DNA can be as degraded as it need be. There's a sensitivity down to 10% mutation load, and actually the assay will determine for you which tumour burden is present. There's a 2.5-day assay from DNA to result, which is similar to most of the microarray platforms out there. Some unique characteristics is that allele-determination is undertaken in the assay prior to hybridization, and also it's the tag region of the MIP and not the genomic DNA that hybridizes to the array, and both of those things are simply designed to get around the poor quality of DNA that you're using. So whole genome copy number can be identified utilising, there's 220,000 SNP probes present on the array. This targets 900 specific cancer genes down to a resolution of 50 kb and then 300 kb across the rest of the genomic backbone. And it's a dynamic range of 50 copies, which means you can have regions of amplification, for example, commonly associated with the MYC oncogene, and you can identify exactly how many copies are present. Very importantly, you can also identify copy number neutral loss of heterozygosity, and this is really an important feature of this assay because other assays out there currently trying to utilise this DNA for stratified medicine are using next generation sequencing technologies, and there's two problems there. One is that next generation sequencing cannot cope with this really poor quality DNA, and another one is that it does not have the ability to identify this copy number neutral LOH. And the thing that I haven't even discussed yet is that you can also identify somatic mutations using this platform. So there are 74 mutations which are targeted on the array, and these include the 9 genes already mentioned as those identified as being most important to target by CRUK. So just to show you an example of results, this patient was referred to us with advanced ovarian cancer. So the two red crosses here in the somatic mutation viewer, which is provided by Affymetrix, shows you that there's 2 mutations that are currently targeted on the assay that are present in this patient's genome. And you can then find out more information in these. So you can see that one of these mutations is in TP53 and the other one in PIK3CA. You can see it gives us here and gives us the HGVS nomenclature associated with that mutation. So in this instance, this was one of our validation patients from phase 2 of the project, and this allowed for accurate confirmation of previously identified mutations. However, clearly there was additional genomic information provided to us, and that was provided in the basis of the copy number data. So this is what the whole genome trace looks like in this patient, and you can see there are many regions of genomic gain and also many regions of genomic loss, extremely complex cancer genome. And if you look at some of these regions in a little bit more detail, you can see there's duplication of the terminal region of chromosome 3. So this region encompasses the oncogene PIK3CA. So we've already said that this patient has a mutation in PIK3CA. The specific mutation this patient has is called H1047R. This is a kinase domain mutation which leads to increased catalytic activity of this oncogene and activation. So therefore increased copy number gain, as you might imagine, is a recurrent finding in patients with PIK3CA mutations. The patient also has deletion of all of chromosome 17p. Chromosome 17p, as I'm sure many of you know, encompasses the tumour suppressor gene TP53. Again, as we've already said, this patient has a TP53 mutation, and therefore showing that this patient also has loss of one allele entirely of 17p shows you that this therefore is a hemizygous mutation that is present in this patient and therefore obviously extremely pathogenic. Also of interest though is this region here. So you can see gains coming up to here. And this patient has massive amplification of this region here. And this amplification is actually including a gene called HER2. So HER2, the human epidermal receptor growth factor 2, overexpression promotes tumorigenesis. Approximately 20% of patients with breast cancer have overexpression of HER2 due to amplification, and actually about 5 to 10% of ovarian cancer patients do also. This is both a prognostic and predictive finding. Prognostic because this is known to be associated with extremely aggressive tumours, and also predictive because it's known that these patients will respond to treatment with a drug known as Herceptin. So because of this finding in breast cancer in 2005, the UK Department of Health recommended testing for all patients with invasive breast cancer for amplification of HER2. Current testing strategies are by immunohistochemistry, followed up by FISH. However, microarray technologies have already been proposed as an adjunctive testing strategy here due to the presence of inaccurate and discordant equivocal HER2 results by immunohistochemistry and FISH testing. And this is really due to the complexity of cancer genomes. So as I've just shown you a couple of slides back, the genome in this patient is extremely complex, many deletions, many duplications. There's lots going on there, not just the amplification of this one gene. So you get artificial skewing of the HER2-KEP17 ratio, so these are the two probes used in FISH, which leads to the expected diploid number of chromosome 17 probe either being increased because there's been centromere amplification or decreased if there's centromere loss. And this frequently leads to confusion in this area. So microarray data clearly improves the accuracy here because it shows you the detailed whole chromosome 17 copy number data, and of course it's showing the whole genome data, leading to a more unambiguous result. And that's me done. I just wanted to say so that Affymetrix microarray systems provide ideal solutions across many different patient pathways within our cohort. I don't have my acknowledgement slide on here, but I would just like to thank the guys at Affymetrix for inviting me to talk today, Dave Curry, who's in the audience, also our field application specialist from Affymetrix, Jeff Spokes, and Ian Pittam. I also want to thank the rest of the guys in my lab, and Deborah and Fabiana in particular, who've run all these technical assays. So thank you much for all of you for listening. Thank you. Yep. Hi there.
So, Zon, so great talk, thank you.
Thank you.
A question, what is the percentage of clinically actionable outcomes from your microarray data for the patient population you studied?
It varies depending on what population you're talking about. Are you talking, for genomic disorders, clinically actionable, we would identify imbalances at least of true clinical significance in approximately 20% of our patients.
Well, as the patients go through your diagnostic odyssey, what is the percentage of treatments that are a result of this microarray platform? That was my question.
The Oncoscan platform,
yes,
or
correct.
the Oncoscan platform, right? Okay. So that's still what we're trying to find out at present. So as I say, what we've done currently is analyse patients where we know there's already mutations present. So phase 2 of this study was looking at patients where we already know that they have these things due to other sequencing technologies, and what we were doing is just showing that the Oncoscan assay can also detect those. And then phase 3, which starts February this year, we'll go on to identify prospective patients, and then I'll be able to answer your question.
Thank you.
Thank you. Hi there.
Hi. We do a lot of microarrays, and I was wondering that as you showed in your one of your earlier slides, a small microdeletion of 24 kb. As in, for anyone who does microarray understands that it's really a nightmare to give a report when you have small deletions or duplications. How significant it is? Like, normally it is understood that less than 200 kb alterations is like, how do you decide that it is significant or not significant? And how do you validate, like redoing the array again, and or if there are known genes within that, you might work on with any other like PCR or RT-PCR. But before that, how do I report this case where I have this question mark that whether it is pathological or not, because the clinician just wants to know whether it is significant for the patient or not.
Absolutely. So the deletion that was 24 kb was in fact in a gene that's an Ullmann morbid gene that's known to be associated with autosomal dominant disease. So even just, I mean, you say down to 200 kb, but if that was the case, then assays such as MLPA and sequencing of genes wouldn't take place, because obviously tiny single base pair rearrangements can lead to pathogenicity. So this patients would have MLPA for identification of this disease as standard. So that would identify these multiple exonic deletions, which clearly would be pathogenic. And it just happened that by microarray, because of the number of probes that are present, we were able to say this patient had an exon 2 to exon 9 deletion, and that's clearly pathogenic because of the gene that it's in. So it really, I mean, just depends on the literature evidence. So if we find, let's say it wasn't in an Ullmann disease gene, let's say that nobody knows what disease this gene causes, then we want to have a look, we want to see what does that protein do, where is it expressed, has it been inherited from mum and dad, and we would look at all of that before we would make our decision as to whether pathogenic or uncertain stroke benign.
And I was wondering that you showed the data on 8 by 60 K array.
Yeah.
I would be a little skeptical to like report a 24 kb microdeletion from this array. I would want to do a higher resolution. Or how, like, what confidence you have in that?
Yeah, I mean, we've identified pathogenic deletions using the 8 by 60K array down to like 1.2 kb because of it. I mean, it very much depends on where your probes are. If your probes are within your exons and you have multiple probes deleted, then each of those adjacent probes is sort of validating each other, as it were, if they're all individually shifted as a deletion.
And second question is that what is the cutoff limit of the mosaicism, mosaic cases that can be detected? Like, is 20%, 30%?
It depends what size the imbalance is. So if you're talking about a whole chromosome, then you can go down like 5-10%. Yeah. But if you're talking about a very small region, then it's going to be bigger than that.
Thanks.
Thank you very much.